Hello, my name is Julian Jacobs, and I work for the Kentucky Department of Fish and Wildlife Center for Mollusk Conservation. Today I'm going to talk to you about the culture of 3 species of freshwater algae used in the propagation of freshwater mussels. The Center for Mollusk Conservation is located in Frankfort, Kentucky on the Elkhorn Creek watershed, and we have facilities for holding fish during infestation. We also have a greenhouse where we have tanks for juvenile culture, as well as 2 facilities, one in Moorhead, Kentucky, at the Monarch Clark Fish Hatchery, and one at Lake Cumberland, which is cage culture, that we use for adult and juvenile holding and grow out. And at the center, we also produce our own algae. We produce our own freshwater algae because it provides the majority of a mussel's nutritional requirements. And we chose the species based on the suitability of its biochemical composition, size, and digestibility. And all 3 of the algae species we grow can provide protein, carbohydrates, and lipids necessary for growth of our juveniles. We currently raise 2 freshwater green algae species, Neochloris oleabundans and Chlorella soriokiana, as well as a freshwater diatom, Phaeodactylum tricornutum. So here's a microscopic view of our Neo, and it's usually about 3.2 to 3.5 microns in size, so it's easily digestible and easily to take it in by our smallest juveniles. And our Chlorella is about 2.5 to 2.75 microns, so it's just a little bit smaller than our Neo. And both of these species, you can see here on the slide, look very similar to each other, and they're very hard to tell apart, except for the Neo at the beginning stages is just slightly bigger. And the Chlorella, if you're looking closely under a microscope, it has a C-shaped nucleus. And both of these species were chosen because they have a high lipid content as well as essential fatty acids and amino acids and high protein. The PT that we grow is about 19 microns in length, but only about 3 microns in width. So it's still about the same size and width as the 2 green species that we grow. But we chose the pinnate form that you see because of its size and shape versus the more football shaped form that it can come in, because the football shaped would clump together when harvested and become very hard to eat because then the size is significantly larger. And PT is high in proteins and lipids. One of the main factors to consider when producing freshwater algae is to always use clean, sterile culture equipment and techniques. To do that, we have a Barnstead unit, which takes RO water and filters it even further to give us ultra pure water to use. A dishwasher that uses high temperature water and steam to clean our glassware. Then we take our clean glassware and add in our ozoned well water and autoclave it at 121 degrees Celsius for under pressure. Then we also utilize a biosafety cabinet that uses UV sanitation for all the surfaces, as well as constantly filtering HEPA filtered air throughout the unit. Then we use our pipettes as well as individually packaged sterile transfer pipettes to give us the right amount of algae and nutrients. Part of our algae culture is to use sterile nutrient preparation and use. So we use Gilliard's F2 Part A and B, as well as essential nutrients 1 and 2, which can be found in the U.S. Fish and Wildlife Training Manual. And these nutrient combinations include boric acid, magnesium sulfate, potassium phosphate, sodium nitrate, and calcium chloride, and they were just deemed as too small levels or not available in Part A and B, but essential for growth of algae. And our metasilicates are used for our diatom. And essential nutrients 1 and 2 and metasilicates can be autoclaved to sterilize, whereas Part A and B have to be sterile filtered because of the vitamins. And all of our algae is kept in an incubator or an environmental chamber at the lower generations. And I kept it with 24-hour lights under sterile air, which is just an air pump with 2 layers of 0.2 micron filters that pump clean air into our algae to keep them stirred up. And at the higher stages, the Neo and Chlorella are grown at room temperature with sterile air mixed with CO2, and and the PT is still grown at just 22 degrees Celsius with sterile air. So the very first stage of algae culture is our slants. We purchase our slants from the UTEX culture collection of algae, and we take that slant that we purchase and put it onto our own slants that we make using Dipco auger. And you can transfer one slant that is nice and dark, take a few cells from it, and transfer it to a new slant and help keep the generation going for a few months. Once we have a slant that has a good culture of algae growing on it, nice and dark, we can transfer the slant to a flask of sterile water. And to do that, we use these sterile inoculation loops to take a few cells from a dark slant into a flask of sterile water. And we could produce, with just a few little cells, it will start growing under perfect conditions into a nice dark green or nice dark brown. We start at the lower level with our Neo, or with our PT, because it takes a lot longer for it to grow. So we can start with a 50 ml flask. It can take a few weeks to a month, whereas our Neo Chlorella can take just about 5 to 7 days with 175 mils of water. After a certain growth period, you get that nice dark color we're looking for. We can check for sterility and exenic conditions under a microscope. Sterility can be very easy to assess under a microscope. And even if there is some level of contamination, depending on the level and the density of the actual algae cells still growing, a culture can overcome a contaminant within a generation or two. So just an example, if I were to see a flask that had this type of bacteria, we can see the rod-shaped bacteria, and that seems to be all we can see, and there are no algae cells growing. So I would deem this unusable because the algae will not grow any darker than it may or may not already be. Whereas if you have this culture, there's still some bacteria to be seen, but the algae cells seem to be very dense, and there are actually some of different sizes showing that the algae is in active growth conditions. So that culture we can then utilize. So here's an example of some other contamination that we can see. We have some filamentous fungus and some other types of fungus, so you can just see the fungal cells. And then you can get euglena, you can get other types of green algae come into the diatoms and just take over the culture. So once you have a good flask growing, it takes, at that stage, you can utilize a 10% dilution from one stage to the next. And the PT usually takes about 6 to 9 days to reach the nice dark brown that we're looking for, whereas the green algae both take about 3 to 4 days to reach the correct density. And so here is just an example of showing, take a transfer pipette and take about 10% of the volume into the next stage. So here, as you can see, there are different stages of algae that we can grow throughout. Starting usually from the slant, in most cultures we go up to a 30-liter or a 5-gallon carboy. And here you can see them growing on the shelf at the different stages of growth. And from there we can either harvest to get the algae that we need or bump it up into a 450 or 100-liter tank to give us even more algae to harvest in about 3 or 4 days. And we always restart in the lab, we restart a 500 ml flask to keep the generations going so that we always have fresh algae that we can use. And at the 30-gallon and all the way up through the 450-liter or 30-liter all the way up to the 450-liter stages, we utilize sterile air but as well as CO2 at room temperature. And the CO2 just helps the green algae regulate the pH because the water pH will go down, and that just aids in faster growth of our green algae. So the culture of PT is very similar to our two green algae since we start from the slants, but it just has a few extra steps because we go from a 500 ml flask to a 2-liter, and then we go to 12 liters or a 3-gallon carboy. And from there, we can either harvest or add it to a 100-liter tank to give us even more of a harvest, or we also utilize a 1,000-liter industrial plankton photobioreactor. And this is just an ultra-controlled unit that adds nutrients and water as we need it. And that just gives us a massive harvest. And for PT, we keep the 500 ml and the 2-liter cultures going to always have a backup culture. And this is just another slide showing how we take 10% of our original culture and put it into the next stage to give us the newest culture of algae. Once the appropriate density is reached, We harvest our algae using either a 300-liter cream separator to give us a paste or a 750-liter US Filter Max algae centrifuge, which give us a highly concentrated algae water. And these both just help remove excess water and fertilizer. And then we take it back in to a 8-liter centrifuge and centrifuge it down to give us our algae paste. And that helps us remove even more of the bacteria and fertilizers that may be in the water. Then we reconstitute our paste 3 times with water to give us the algae that we need to use. And we keep that refrigerated for 5 to 7 days, and we use it every day in a 2-to-1 ratio with commercially available marine diet. So our freshwater algae is then fed along with reed mariculture's nano, selfish diet, and TP, and fed at a fixed ratio with our together to feed our juveniles. These are fed every day by mixing the correct amounts based on the number of trays that we have running in the greenhouse, and centrifuging them all again to remove marine salts and any other bacteria or fertilizers present in the water. And our stagnant trays that we use only get about 1 gram total of algae per day when they're very young juveniles. We also add a probiotic to the trays to promote healthy bacteria and biobarrels to help reduce the buildup of ammonia in our trays to keep good culture conditions. So that is just a little bit of information on how we grow our 3 freshwater algae species and why. But none of this work can be done alone. We just have to acknowledge some of our staff as well as some of the other field assistance that we get from different agencies and our funding sources that we get to help run our facility, as well as our families that help us keep working. If you have any questions, you can submit it to the board during the conference, and I'll try to get back to you with any answers that I can provide. And thank you for listening.